nicely, the overview is actually complete. So all our role is already played. So essentially the innings is over. We can now
No. It is not. The director of the Genomics Institute has to talk about variants.
Right. So yeah, that you didn't touch upon much. That's true.
Yeah.
The implications of the variations. But you have already toned down the effect of variations. So I think Gagandeep is there.
I don't see her there.
Okay, I booted him off now.
Okay.
I think I booted him off, but maybe not. Now Saumitra may be sharing.
Which one? I'm not sharing.
Okay, anyway.
So, Shashank, how to go about? Do you have questions that we should answer?
Yeah, so the questions are coming in the chat box. So let's wait for Cherry and Sangamitra to join, but then let's start. So
yeah,
Kumar
please start.
is asking,
I'll try to reach out to them again so they can
yeah,
join.
so Kumar is asking, recovery rate is high and death rate is low in India. Is it because of some natural cross protection among Indians? Can the government take this as an achievement? Well, I will not comment on what the government can do or cannot do or should not do. But there is certainly evidence, and recently Shekhar Mande has published a paper with some people at the Mathematical Sciences Institute in Chennai, which has looked at the countries, various countries, and has infectious diseases in those countries and correlated that to death rate in COVID. And it appears that if you are exposed to other infectious diseases, which we are in India, then you have some protection from COVID. This could possibly be because of innate immunity. So that's possibly it. So, but it's not really just in India. As I also showed, the death rate is low all across South Asia, all across Southeast Asia, Africa. So it's not really an Indian phenomenon.
But Shahid, I think recently Professor Partha Majumdar pointed out the people who are taking flu shot, somehow if they get COVID, their severity is not to the extent of the people who are not taking flu shot.
Right. So it's the same thing, Saumitra. People who take flu shots, the flu vaccine. There are two types. Most of the flu vaccine that you get in India is a killed vaccine, whole virus vaccine. So it will also set off innate immunity, at least to some extent.
Right.
Could I add to this, Shahid?
Yes,
so great, really nice overview. And I think this is a really interesting question. And I think you put it really well, Shahid, that there is a certain myth that the death rate in India is somehow significantly lower than that in the rest of the world. I think you also made the more nuanced comparison that the death rate in India may be lower when compared particularly perhaps to America and to Europe, but not necessarily to the rest of the world. And there is a lot of discussion, exactly like you said, on how that could potentially be explained. And I would like to concur, and we've been thinking about this and working in the area of BCG and innate immunity. We do think that the potential that of exposure to environmental and gut microbes being very different between that of in a country that is endemic for NTMs, non-tuberculous mycobacteria, Mycobacterium tuberculosis, and other gut fungal pathogens as well, microbes, would be different, would set a different innate repertoire in our system compared to those countries where these microbes are not endemic, and consequently that the innate initial response, particularly the type 1 interferon response, which I'm looking forward to hearing, there's going to be a review on that, may well be very different. This is a testable hypothesis and one that I think is important and needs to be formally verified in a comparative analysis between populations where the death rates are different versus India. And there is a call out now, which we are looking at very carefully on just that, between South Asians in England versus Indians in India. So I concur, but the mechanisms of how the innate response is behaving is something that I think is important and we should test. So I concur broadly with what you're saying, Shahid. Yeah.
Thanks, Anna. Thanks. So Kumar has another question, and I will.
Shahid, how accurate is the data that is coming out of the countries which are showing less death rate than that of India?
Well, I mean, you could say the same thing about India. How accurate is the death rate in India?
You could definitely.
You know,
India is also,
death is kind of hard to miss.
Yeah.
Let's assume that the data everywhere is equally good or equally bad. Let's not be selective about it. So it's based on that. Saumitra, take this question from Kumar. He says the G-clade virus is taking much attention, and I assume he means the D614G virus. Taking so much attention worldwide, probably occupies more than 80% of sequenced genomes. Did we discuss about this enough today? I don't know about that, but
yes, I did actually. Kumar, you might have missed my talk only, because in my talk I highlighted on D614G and actually also tried to show some molecular basis why its affinity towards SCE2 is increased. Actually, we did a study and published it in May, if I am not wrong, from India only. And we showed by computational studies, Srinivasan modeled it. And he showed that because of this mutation, when you are removing aspartic acid and bringing in glycine, it's actually bringing in more flexibility into the structure, in the conformation of that domain. And that, it has actually two structures. One is before it binds to the receptor, and the other one is after it binds to the receptor. Now, because of this conformational flexibility, so it can now acquire as I said, that metastable pre-fusion and the post-fusion, that actually changes with the D614G. Now, because it is highly flexible and supports the wobbling nature of the interprotomeric hydrogen bond between the D614G and a neighboring, it's a tyrosine residue 859, and that's probably gives the flexibility. So we did discuss about this, but and 79.4% is our, we have our findings suggests from the 1,000 genome that we did it from India. So in India is also the similar situation.
Okay, wonderful. Cherry, welcome to the panel.
I just wanted to update Dr. Saumitra has gone away for an urgent meeting. She will not be able to join.
All right.
I suggest there are other experts also in the meeting right now. If anyone has an urgent point, they can turn on their video audio and pitch in.
Sure.
Thanks.
Sure. But before that happens, Cherry is here, and I was hoping that she can take us through very briefly through the vaccine scenario and answer a very simple question. When are we in India going to get vaccinated?
So it's a tremendously exciting week for vaccines with the Pfizer results, because it proves in principle that a vaccine that is based on the spike protein will succeed. And not just the spike protein, the Pfizer vaccine is actually based on the receptor binding domain of the spike protein. So you have this stabilized pre-fusion structure, but it's really the RBD that is inducing a protective immune response. Now, will the Pfizer vaccine itself be good for us? It's an mRNA vaccine. There aren't such vaccines that have been licensed so far, but all the signals for Pfizer getting an emergency use authorization by the end of the year looks really good. They haven't had significant safety issues. Their vaccine trial is very large. 44,000 participants, of whom 39,000 have received 2 doses. 2 months of safety data will be available by the end of the month. The initial stages showed pretty good binding and neutralizing antibody responses. So things are looking good for one vaccine, which means that things are likely to look good for many other spike protein candidates. Not every single one of them, but a large proportion. Now, if we look at the vaccine landscape in India, we have only one company that is focused on RNA vaccine, but many companies that are focused on spike-based candidates. So given that we have Bharat Biotech's vaccine now getting into phase 3 trials, and we are likely to have at least 4 vaccines that will give us phase 3 readouts by the end of the year. One of those includes the AstraZeneca Serum Institute of India vaccine. If there is efficacy data from AstraZeneca's trials that are taking place in North and South America, the UK, and in Africa, then it's very likely that Serum Institute of India will apply for and get an emergency licensure. In India, we have not had this kind of approach before, but I think it is now being discussed. 1,600 people have been recruited in India for an immunogenicity study. So we will have sufficient data for what is usually a bridging study when we think about vaccine licensure with Indian regulators. So our first vaccine, if AstraZeneca's candidate works, will likely be that. The other vaccines that are heading in that direction are Bharat Biotech and the Gamaleya vaccine.
So, Cherry, I had a question. How concerned are you about the ultra low temperature storage for the Pfizer, for the mRNA vaccines, basically? Because that creates some huge challenges for distribution.
Yeah. This is not a vaccine for us. It's too expensive. RNA vaccines are hard to make because of the stability issues. These are formulated now with lipid nanoparticles. And if a vaccine has to be delivered at -80, it is not a vaccine that can be used by everybody in India. This may wind up being a product that we could think about for rich people in cities who can afford to pay for these vaccines, where such storage facilities can be set up, but it will be a very limited usage in this country. I think given the kind of situation that we have, we should wait for the other vaccines which are better suited to delivery through our systems, rather than going after the first successful product and saying, oh, this is fantastic, now we need to deliver it to everyone. I think that is likely to be a huge financial strain on the country. It neglects other important public health problems. So while this is a success story, I think we should treat this as proof of principle and focus on getting to market vaccines that are more suitable for us.
Okay, I have a question here from Anna, and that's to you, Cherry. She says the Merck vaccine is really exciting, as you said. I think what she means is the Pfizer vaccine, not the Merck vaccine. For the first time that an induced immune response can protect and provide a basis to correlate protective immunity studies. Given this data, why have previous vaccine attempts against coronaviruses been challenging? So she's talking about SARS and MERS.
So there were efforts to make a SARS vaccine. 2 vaccines for SARS went into human clinical trials. Both did phase 1. One was a DNA vaccine and one was a protein construct, but then there was no way of taking them further because there was no disease left. So you could not evaluate them in a clinical scenario. There was no question of getting a clinical readout when you don't have disease. So the SARS vaccine program stopped because of that. Now we have one of the original SARS vaccines that's going to be taken up by BioE. This is the construct that is being made by Vellore, by Peter Hotez and Maria-Elena Bottazzi. So that is going to be used as a vaccine for SARS-CoV-2 because of the cross-reactivity between the 2 spikes. The other MERS vaccines, there were no serious attempts to make a MERS vaccine until CEPI came along. And in fact, the AstraZeneca vaccine actually comes from a MERS program that was funded to the University of Oxford that was repurposed very quickly to become a SARS-CoV-2 program. So I think even with MERS, it's likely that if we have a good platform technology that works, whether that's RNA or adenovector vaccines or anything else, we will see more advances for other coronavirus vaccines now.
Great. There's another question for you from Kumar, but before I go to that, there's a pending question from Usha. And she's asking, other than death rate, are there studies that report differences in sequelae? Anyone wants to take that up on the panel?
Shahid, can I answer that question?
Yeah, please, go ahead.
Based on the viral genome sequencing, if at all only one major finding was the appearance of the G-clad virus. That virus appears to occupy 80% of the worldwide sequence genome simply because it is able to transmit much faster than the other viruses. Otherwise, comparison at the level of severity of the disease, including the death of the infected patients, absolutely no difference between any of the strains so far isolated have been shown.
So
okay, so I think what Usha meant was, and Usha, if you're here, you could chip in and
I'm
say,
very much here. I'm the least of the qualified to speak of animal viruses. I was just talking from the human side because I am a human, and I know that there is a huge amount of genetic diversity in our makeup and our physiology, and therefore, post-infection, the virus might just be the same, regardless of the genetic diversity or the evolution of the virus. But we haven't evolved in the time scale that the virus has evolved. But we already have our evolutionary history, you see. So therefore, the way our cardiac system, our physiology, our Indian set of genes that we have behaves to this virus is likely to be different than the others. So even when we have taken care of the patient and sent them home, how long, how much is this burden? Is it too early to tell what this burden is?
I think Cherry wanted to say something.
Yeah.
Cherry, go ahead.
So we keep talking about the environment shaping both pathogens and hosts and our interactions, right? So I'm going to leave the environment out of it, but I can tell that from the clinical literature, there seems to be two things that we are seeing that were under-recognized. One is the extent to which autoimmunity plays a role. So earlier we were thinking cytokine storms, this is what is happening. There are coagulopathies all over the place. This much was sort of understood. What is emerging recently is the importance of autoimmunity. Now, when we say that, then looking at autoimmune diseases across the human spectrum, you can start to see patterns. And it's likely that if we begin to think about the more chronic sequelae of SARS-CoV-2, it is likely to be linked to people who have different genetic backgrounds. To some extent, environment is likely to matter, but I think it's the sequelae we will find are probably going to be associated with the host genome. The other thing is with viruses, we have signals that the many viruses tend to be associated with particular types of receptors. And we've had odd signals coming up saying this blood group is protected and that blood group is more susceptible. Now, this is something, again, is not first for SARS-CoV-2. We've seen this with a lot of enteric viruses, for example. So when you think about mucosal infections, it is antigens that are expressed on the surface that are genetically determined that make a difference to the uptake of the virus. So I think this is an area that really requires much greater study.
Thanks.
Cherry, there's another question for you from Balaji. Can you comment on the role of haplotype polymorphism of patients belonging to different sections of society?
No, I can't. Don't know the first thing about it.
Yeah. Okay, so we leave it there. Shashank is asking, and maybe one of the other panel members can chip in here, what can be done to minimize the regulatory hurdles, facilitate reagent sharing and collaborations during next pandemic? This has been a major roadblock this time in engaging the research community.
Solution, but definitely Cherry and me both will share our experience of not getting samples from or even other signals from ICMR in time. So this we faced because I think we could move much faster to even for sequencing to get the samples from the hospitals. I have faced problem that the hospitals authorities are ready to share, but they are not getting the signals from ICMR. So I think Balaji, being Balaji, didn't really mention, it's a regulatory, he has mentioned.
Oh, this is not Balaji's question, this is Shashank's question.
Oh, this is Shashank is now the junior to that. So I'm just saying, essentially, we all pray that I think in the next episode, somehow we mobilize these hurdles so that the scientists get an access to the reagents and samples to really address the problem. I think it took a long time for us to convince the authority to have an access to that before we started acting on it.
Yeah, so we can all hope and pray for a more engaged ICMR.
I think the other thing that we can do is, I think if all the governments, what I mean is the state and the central government, they should really decide their protocol how to implement a lockdown and close down the institutions. I think one of the major deficiencies that most of the research institutions felt was that all of the students had already been sent home. And so even if you wanted to do something, you wouldn't be able to do because you don't have your students with you.
Saumitra, that was not a CIDR question.
Right. It is not a CIDR question. That's what Balaji is saying. No, but it is true. But somehow, I'm in a small institute, and we were actually somehow we kept motivated the students, and they stayed back in the hostel. And all the students worked during the pandemic. They didn't really worry about the lockdown. But I made sure they are safe in the campus. But for a big institute, it's definitely an administration problem, and we can't really expect them to take this challenge.
So, I hear that institutions had to report on a daily basis what they did that day.
Yes.
For COVID. Was that a hindrance, or was that helpful?
Why do you think it would be helpful?
You never know. Some people may like it.
So helpful in the sense you don't lose your focus, because you need to report to the authority that you have actually how many tests you have done the whole day, or how many sequence you are done. And if there's a pressure, it's like an indirect pressure on you to perform. But somehow at some point, I mean, all negative, but there is 1% positive is essentially that keeps the people, even under this demanding situation, they really perform. Otherwise, knowing the human psychology and the part of India I'm staying now, may not really worry about it too much.
Shahid, can I ask a question to Saumitra? Is it possible on variants?
Yes. Let me go through one question, and after that, Anna, please. So Kumar has been asking this question, and he repeated it.
BCG.
Can anyone on the panel talk about a Nature report about unproven COVID drugs in India? So
I'm quoted in that. I'm quoted
okay, so
as
then
the
you're
only
the best person to talk about it.
It just came out today. Today's issue, it came out.
Yeah. So basically, the question was around the. So it wasn't about the whole Ayush scenario, which is another point of discussion that people have for proven and unproven drugs. This was around the use of drugs that are antivirals, but not necessarily proven to work in SARS-CoV-2. So, for example, the small study that Biocon did, the trials from Glenmark and others that have used favipiravir, etc. So the argument there was these have not been evaluated for the purpose for which they are intended, and therefore India is not going in the right direction. So basically, they're right in that doing small trials is unhelpful. You do a trial. So the question was, should the regulators be blocking this drug? The drug is one that already is licensed and is marketed in the country. The company does a small study that shows that it works in a 100, 150, 200 patients, and then goes to the regulator to say that we would like this to be an additional indication for this, or we want to do the next stage of trial. Should the regulator stop them or give them an ability to modify their label? So to me, the question was around equipoise. What sort of situation do you decide that a drug should be used in this setting or not be used in this setting? When the entire country is using, favipiravir, for example, then how do you achieve equipoise when you want to set up to do a clinical trial? To me, these are very difficult issues. I think it's one where the regulator is not responsible, but as an academic community and as the government, we should be thinking about ways of better evaluating therapeutics. I think we need networks of clinical trial sites. we need centrally coordinated clinical trials to be done to show us whether these drugs work or not. It's not the regulator's responsibility to do that.
Okay, Anna, go ahead, ask your question.
Saumitra, first of all, congratulations on your 1,000 genome, your project, and I read the paper with interest. So my question is that, are you able to link the sequencing to track and trace? So you've kind of given some information in your data. I missed your talk today. It was really early, but I missed your talk. You gave some indication of the points of entry of the virus into India. That is the number of points of entry in a kind of inferred way from your data. Are you able to move this into more real-time track and trace to understand the extent of real-time emergence of mutations within the community? Are you able to do that? And because that would give us an idea of to what extent it goes back to sequelae and all that, to what extent the viral dissemination in India is actually having an impact on disease.
Right. So actually, you are absolutely correct. This is important. But what we faced, it is very few hospitals actually shared information about, or even where from we collected the samples, shared information about the patient when, they might have shared with us like what is their virus load or, what is their severity. But they didn't really share the information that when they were coming, whether they actually had a history of travel. All these informations was actually challenging. So where from we collected NIBMG, remember it was actually 5 different centers we conducted it. Whatever we collected, we made sure that we had an access to those 500 samples, but the rest 500, I'm still struggling to get those track and trace information. So then only we will have a better. But I think what we are now as a government regulatory, we are just trying to see whether if we can impose that for the next pandemic, this becomes an organized study where people will be, from the beginning, they will be alert and then keep the records in place so that it can be shared. Because a lot of times, because of this panic, initially, particularly March, April, it's very difficult to get any information in March and April because people were very, very shaken up, and then they just somehow treated the patient. They didn't know that they themselves will survive. All this kind of a fear factor work. So I can't blame them. So I think this is something which we need to plan for future, that we will be much organized to handle such an epidemic, so that we have all these informations in place and something like that can be shared.
Yeah, thank you.
Shashank, how are we doing with time?
Sahid,
just a minute.
Sahid, can I add?
Can you please wait? Shashank.
Yeah, we have 5 more minutes. They have already taken up the coffee break,
Okay.
which was virtual.
All right. There's a question by Dinesh Gupta. What should government do now to slow down the peak? Well, we have already gone past the peak. So I guess what you mean is slow down the outbreak. Anyone wants to take that one?
Cherry.
Yeah, Gagandeep, yeah.
So I don't think we should be complacent. I think Shahid, you're very well placed to answer this, but I think that given our population and the way we live, we still have plenty of susceptible people out there. In many places, we are getting to 30–40% seroprevalence, which should be slowing the virus down by itself. So what I'm not sure about is whether this declining that we are seeing now will turn us into a situation that is more like South Africa, where you go past the peak, but then you continue with a fair number of cases for a fairly long time. rather than coming down at the same pace at which we went up. And then, of course, the worry about second peaks is also there, but the amplitude might be lesser.
So yeah, I think the attitudes are going to matter, and people are already in a COVID fatigue. That's on one end. The other end, we are into a festive season. We have just gone through an election. Wait for 2 more weeks and see where this is taking us. I think that will be a very good indicator of whether things are slowing down or whether things are going to be sort of stabilizing at a point higher than baseline. Hard to call at this point.
Maybe I can have the last question. How do you sort of counter this fatigue point? Because I think that's really important thing. If you can have people continue to follow the safety measures, I think as you mentioned, the mask and other thing, that is going to be a real help.
It will be. And I heard a very interesting comment the other day. When you pick up your phone to call somebody, Amitabh Bachchan comes online and tells you to wear a mask. I'm sure many of you have heard that. So someone was saying, why is this guy telling me to wear a mask? He got infected. His whole family got infected.
That's why.
What authority does he have to tell me to wear a mask? So I think what this says is that communication has to be proper. Just because Amitabh Bachchan has a good voice doesn't mean he's going to be a good ambassador for everything. So what I'm trying to say is perceptions matter. And we have to be clearly communicating the right message so that it reaches people correctly. Probably that's going to do, that's going to help us the most.
Management is not really the exact science here, and I think that's really the key point. How do you manage the situation is really an issue.
Shashank has a question about...
sorry. I somehow don't have this hand raise tool. So I suppose that's a feature of this live event. Yeah, human behavior and psychology is really being tested over here. So, it's difficult to change things. The older you are, the more difficult it is. And, people are set in their ways. So really, it is communicating to them how, it is their carelessness affects them and their immediate family. Maybe that's the only way it'll go out. Because clearly people haven't understood that wearing a mask it's required that about 70 or at least 80% of the population should be masked in order for there to be any effect. So that message hasn't...
It is effective even less number of people are wearing it. I mean, for those people, it is effective.
No, when only 20%, 30% are wearing it, then it's not much effective at all. Yeah, it is effective for the person who's wearing it to some extent.
As well as people who are around them. People who are around them.
Yes, so I think it's really difficult. We have to make them understand and make their life easier to say that, okay, yes, you can go shopping, but this is the only way by which you can go shopping. This is the only way by which you can travel. But if you don't, comply, then the consequences of that are what people need to understand. The communication of the consequences of irresponsibility is the problem.
Well, at this point, Delhi's Chowdi Bazaar and Bihar election rallies don't look very different at all. People are not wearing masks in either places.
Right.
Shashank has a question on how to use COVID-19 diagnostic labs in inter-pandemic period.
Yeah.
So he is assuming that the next pandemic is just around the corner.
So
Well,
yeah,
I would say do some really good virology research in the inter-pandemic period.
I think for a country like India, there will always be infectious diseases which will require, it may not be a pandemic, there might be something which is a local outbreak or one or the other, and being nimble on the feet and, like the Oxford people did, is to take something which was MERS and convert it, something like that, being nimble.
Okay, so
I think Shashank has the last word now.
Yes, I think we have had a good discussion.
We have assumed the coffee break also, and we are running over time. So thank you so much, all of you, for joining. This was
Thank you, Shashank.
More serious chit chat than like a fun chit chat than a very serious discussion. But I guess everybody was